The refractive media of the eye. The refractive apparatus of eye comprises the cornea, aqueous humor, lens and vitreous body. About two-thirds of a refraction of light takes place by front surface of the cornea at the junction of air and corneal epithelium. Cornea, we have already studied about this in previous videos. Now we'll see about aqueous humor. It fills up the anterior and posterior chambers of eye and is compressed of approximately protein-free plasma. It carries glucose, amino acids, some hyaluronic acid, high concentration of vitamin C and mediates the respiratory gases. The aqueous humor is formed by active process and by diffusion from the capillaries of ciliary processes and is initially collected in the posterior chamber. Hence, it appears in the anterior chamber through the pupil. Since the pupil is closely applied to the anterior surface of lens, it permits the fluid to flow from posterior to anterior chambers but not in reverse direction. On reaching the iridocorneal angle, the fluids find its way in sinus venous sclerae, which is known as canal of Schlemm, through the endothelial lined spaces of the trabecular tissue, which is known as spaces of Fontana. Finally, it is drained into anterior ciliary veins through the aqueous vein. Some amount of the fluid is also absorbed through the anterior surface of iris into the iridial venous plexus. Functions of aqueous humor. It provides nutrition to the cornea and lens. It maintains the intraocular pressure. The normal pressure is about 15 to 20 mm of Hg and is calculated by tonometry from the measurement of impressibility of the anesthetized cornea. Tonometry was kept on the cornea when it is anesthetized. Without anesthetizing the cornea, it is not possible to apply because cornea is hugely supplied by the nerve endings. Now we'll see about the lens. The lens is a transparent biconvex flexible body that intervenes between the iris in front and vitreous body behind. It presents anterior and posterior surface separated by rounded border, the equator. The posterior surface is more convex than anterior and rests on hyaloid fossa of the vitreous body. The central points of both surfaces are known as poles and the line joining anterior and posterior poles from the axis of lens. It is anchored to the ciliary body by the zonular fibers which blend with the lens capsule around the equator and forms a suspensory ligament of lens. The average diameter of lens is about 1 centimeter. It contributes about 15 dioptries. The dioptries is nothing but equivalent to what you call as a power of lens, what we call, okay, 15 dioptries to the total of 58 dioptric power of the eye. The advantage of the lens over other refractive media is that it can change its dioptric power for the near or distance vision by adjusting the curvature of its anterior surface. The lens absorbs much of the ultraviolet light. With the age, lens becomes increasingly yellow and harder. As a result, the power of accommodation for near vision is lessened, producing presbyopsia. This defect may be corrected by the use of convex spectacles. An opacity of lens is known as cataract. Structure of lens: it consists of capsule, anterior epithelium and lens fibers. Lens capsule: it is a transparent elastic basement membrane with an abundance of reticular fibers and envelopes the entire lens. It is formed by the epithelial lens cells, contains sulfated glycosaminoglycans and stains brilliantly by PAS technique. At the equator of the lens, the capsule is fused with the fibers of a ciliary zonule. Anterior epithelium: beneath the capsule, the anterior surface of lens is lined by a single layer of low cuboidal epithelium. Towards the equator of the lens, the epithelial cells elongate and differentiates into lens fibers, which turns meridionally and forms the bulk of the lens substance. Let us see about lens fibers. The laminated structure of lens is due to continuous addition of fibers in the region of equator, and this process goes on throughout life. During the transformation of lens cells into lens fibers, the old fibers in the center lose their nuclei and the new fibers at the periphery possess a flattened nuclei. Hence, the harder central part of the lens is known as nucleus and the peripheral softer part forms the cortex. The nucleated and non-nucleated lens fibers remains alive and contain longitudinally disposed microtubules and characteristic crystalline proteins. In absence of nuclei, protein synthesis of fiber is maintained by long-lived mRNA. Number of lens fibers in adult is about 2000. On cross section, each fiber is hexagonal prism. Now we'll see about vitrous body. It is a transparent gelatinous mass which fills up the posterior 4/5 of the eyeball. It is composed of 99% water with some salts and contains meshwork of collagenous fibers and mucopolysaccharides and hyaluronic acid. A narrow hyaloid canal extends forwards through the body from optic disc to the center of the posterior surface of the lens. The canal is occupied in fetal life by hyaloid artery, a continuation of the central artery of retina, which normally disappears about 6 weeks before birth. The vitreous body is enveloped by delicate and transparent hyaloid membrane, which is attached to the ciliary epithelium and ciliary processes and to the margin of optic disc. Anteriorly, the membrane forms a depression, the hyaloid fossa, upon which rests the posterior surface of the lens. In front of ora serrata, the hyaloid membrane is thickened by introduction of radial fibers to form ciliary zonule. Now let us discuss about some applied aspect regarding these topics today. The first one will be glaucoma. The obliteration of a iridocorneal angle prevents the absorption of the aqueous. The consequent rise of intraocular tension is known as a glaucoma. When the condition persists, it produces cupping of optic disc due to outward bulging of the lamina cribrosa and culminates into blindness. Next one will be like the iridio-corneal angle can be examined in vivo by special instrument, the gonioscope, which comprises the corneal microscope and special contact lens. The movement of specks like flittering flies is sometimes seen in the vitreous body as musky violet eyes. Possibly the specks are formed due to the red blood corpuscles in the vitreous body.